So my name is Anouk Rijs, and I recently started as a professor at the Vrije Universiteit in Amsterdam, where we are in the analytical chemistry department, and we use mass spectrometry combined with spectroscopy, and we do this to target peptide aggregation. And so the first question is, why do we want to do this? What is so interesting? Well, where are these aggregates? So let's have a look for that. So peptide aggregation or protein aggregation is a very important part of human health. And as you can see in this picture over here, there are a list of diseases: Alzheimer's, Parkinson's, ALS, Huntington's disease, that are all related to this peptide and protein aggregation. So you can imagine that we really want to understand what's happening when proteins are aggregating, because they are related to many of these neurodegenerative diseases. So then is the question, what is aggregation? So what does the word aggregation mean? Well, it's clumping or clustering of proteins together. So they change their shape, their form, and they stick together like tape, which you put on top of each other. So this really affects the function of the protein. Well, where does it happen? Well, as you can see, it's related to these neurodegenerative diseases. So it's not surprising that this takes place in our brains and at the level of the neurons. So one of the diseases that we are working on a very broad scale is Parkinson's disease. And it's related to the protein alpha-synuclein, which is depicted over here. It is a very disordered protein, so it doesn't have really a native structure and not even a real function in our body, but we do have it. And when it's unfolded, it can stuck together. So we have many of these proteins aggregating or clumping together that actually make this kind of plaques that are in our brains when people are diagnosed with diseases as Parkinson's. So typically the aggregation mechanism is very schematically depicted over here. So you always see these two bars, which means that a peptide or a protein is unfolded and formed a so-called Peter sheet structure, which is indicated as these linear bars. It's a very extended structure. And when they aggregate, you get that these units are stacking together like a ladder-like structure, as you see in this very schematic picture over here. Well, the question is, how are we going to study this? So we know that it's important, we know what it is, but what are the methods that we use to study and what has been done so far? So to study peptide aggregation is a big challenge. So what has been done so far is that a lot of things is known on the proteins that are related to these diseases. So you could see in this previous slide, they have the different colors. They are different proteins that are related to these diseases. So these proteins, they have been identified. The structure have been tried to elucidate, for example, by crystallography, a method that really depicts the overall structure. So that's on the one side. And on the other side, a lot is done on the final results of this aggregation. So they form plaques in our brains, as you see on this image on the right. So they are investigated by imaging techniques where slices of brains are studied, or by microscopy techniques where you actually really see this long fibrils, so this long range of peptides, proteins stacked together in this ladder-like structure. So on both sides of the process, a lot is known. But the problem is that the most toxic species is the one that's actually in between these 2 points. They actually, when it starts to unfold and aggregate, and these early, what we call oligomers, they are the most toxic for us. So, of course, that is the ones that we want to understand, to study, and why, what's the structure, what can we learn about them? Well, this is a very schematic presentation of what's going on. So you have what we call the on pathway, so the things that lie on this curve. So that's the aggregation, when you go from monomer to dimer to tetramer, etc., and you have this ladder-like row. But you can also have partly unfolded proteins stacked together, So they can go off pathway, they can fragment again and become smaller again. So it's a very complex problem. So if you would study this, you have like a soup of many different structures, many different sizes of aggregates that started at different time points. So it's really complicated if you want to say, well, we want to pick out one of these structures, look what it is, and pick out the next. Because whatever technique we use, if we measure it together, we just get the signature of this whole network. So that's a big, big challenge. And before we go into see how we solve this big challenge, I want to inform you that peptide aggregation or protein aggregation, well, we study it for neurodegenerative diseases, but it's also very important for other methods, like you get the self-assembly of peptidomatics, so functionalized peptides that can form interesting biomaterials. They can so mimics can form, and they can form this coacephate, so we can study droplets inside droplets. So meaning that you can, for example, build artificial cells. They are self-synthesizing molecules, so when you bring them together, they rapidly form a macromolecular structure by themselves. So this whole process that we are talking about, well, we focus mainly on the neurodegenerative diseases, but the knowledge is transferred to other organic chemistry disciplines as well. So just to come back here, so we have this very complex network where we want to pick out individual structures to look at them to actually map this aggregation pathway. So this is what we want to do. So that's one part. We want to understand the molecular mechanism. So really look at individual parts. When we've done that, we know what it looks like. So we also want to understand and understand if we can stop this aggregation process. So can we see that the therapeutics that are used now to, for example, slow down the disease progression, can we use them earlier on so that they do, for example, more? So we want to stop this process. So this is the more fundamental part of the research. We also are working on the more applied part. So where we do actually the diagnosis of Parkinson's. So it's now done by looking at the by external symptoms like the motor symptoms, so the shakiness, as you know. So there's a lot of tests, but there's not a molecular biomarker that actually tells you really that you have this disease and not another brain-related disease. So what we also work together with the group in Manchester to work if we can find biomarkers that actually tells you that you have Parkinson's and ideally at what stage you are. So for this, we need to develop a lot of methods, experimental methods, and that's what we're doing at this moment at the VU. So we combine, and that's why we call it 4D, different methods together to actually study this complex problem. And I will tell you today what do we combine and how does it help us to bring the answers. So first look at this molecular mechanism, this understanding of the molecular mechanism. So if you want to understand the molecular mechanism, what does it mean? Well, it means that you want to know these initial steps in the beginning, so in the early stage of our curve, that results and forms these fibrils. And we want to identify intermediates along this pathway that are very important, that, for example, are key for fast development or actually are points where the growth is slowing down. And of course, we have this on pathway, but we also want to see what are the off pathway components and can we direct the aggregation towards the off pathway, for example. So you want to see the difference between on and off pathway and why is one stimulating the plaque formation and fibril formation so much and the other one is more safe. And then, of course, we compare this to functional aggregation. So one of the examples I showed before. So how is it different? So why is aggregation of one protein toxic and the other one actually are useful? For example, hormones are found to be stored as fibrils as well in our body. So why is it on one hand okay and on the other hand not? And of course, most importantly, can we control this process? So I already told you we have this very complex soup of structures and we want to pick out one, look at them and pick out the next. So how do you find the structure where you're looking for? Well, this reminded me of a book I used to have as a child, so I don't know if you know it. So it's called Where is Waldo? And depending where you're from, it might have been Wally or Walter or any other of these names. And you had this cartoon where you had full of people, and you had to find this person like this. So you really, here you have to find the one structure that is in structure, Waldo, that's important for the aggregation, well, for your game in here. So this is a similar problem as we have now. So we want to find this one structure, look at it, find the other one, and see why it's the same or different, and what is it? Is it Waldo or is it just a random person? So this is another way of looking at it. So on the x-axis, we have the number of aggregates, where in the yellow arrows say when we have this nice aggregation, and the blue, well, they look like kind of sausage-like structures. The folded ones are the ones that are off pathway. And of course, we can have one of them together, two of them together, 4, 10, 20. So we need a method to actually select a specific size. So we have different sizes. And then, for example, within the NS4, we have, besides different sizes, we also have different shapes when we are on pathway or off pathway. And then within them, we have interactions that help us to understand what Waldo or what our structure look like. So different sizes you can very well characterize with mass spectrometry. And mass spectrometry is a technique to just simply weigh your molecule. And of course, the molecule, if you have 4 units together, is twice as heavy as you would have 2 units together. So you can use mass spectrometry, very simple, to select a specific mass, you know how heavy it is, and then you can pick your aggregate. So then you are, for example, in the NS4 channel. Then you want to select a different shape. So for example, you're interested in the one that I highlight, and you can use this by techniques either with UV light or ion mobility, which are both techniques that select a specific conformation, so that separates these 3 structures because they have different properties. And we will look into it a little bit more in detail later on. And then we use spectroscopy. So we pick out this one specific structure, and then we measure its infrared spectrum. So rather than of the full ensemble, we just select one, then measure its spectrum, and then we have a fingerprint of this structure, which we can then read out and understand what it looks like. So this is what we do. Very briefly on the different shape part. So we have 2 ways. We can do UV probing. I will not go into a lot of detail, but what you can do is when we are at in cold temperatures, we have very high resolution in our UV signal. The UV signal between conformer A, so the sausage arrow type combination or the 2 arrows, this UV excitation is slightly different. So the excitation of energy, so the UV color of the UV that you need for to actually probe the A conformer over the B conformer is slightly different. So by tuning, so if we use a laser, we can select one of the conformations. The other one is ion mobility. And that is actually what we use at the moment in the lab, is that we have an electric field. So it's inside our mass spectrometer. So we have an electric field that goes from left to right. Our 2 or 3 conformations, they go inside this ion mobility cell. And there's an electric field that pulls them through the cell. But the cell is filled with helium. So you have a lot of atoms flying around and bouncing against our protein. And of course, you can imagine when you're small and little and folded, you have less interactions with these bouncing helium atoms than when you're tall and extended, because your surface is way larger. So you have more collisions with helium, so you're more slowed down. So if you then look at time, then the small folded one will arrive sooner than the large one. Well, you can see that the same when we have two skiers. One is very good and is nice and bended. The other one is a new first-year skier and stand up straight and is much more careful. So when they go down the slope, the folded, the small one, the faster, it will be much faster than the extended one. And of course, this will result in the same type of separation. So that's what we will use. So you can see if you are larger, you go slower, and if you're small, you go faster. So that actually helps us very well to sort things out. So if you look back at conformer A and B, when you have two extended ones, or you have one that's more folded around. So here, B will arrive later than A. So these are the techniques to do mass selection and then shape selection. For those who are still looking at the picture of where is Waldo, so here he is. So that's also, if you were still looking at that rather than the techniques that I was explaining, then you have your answer right now. So maybe you find him, maybe not. So we will do this in our lab to combine these techniques that I mentioned. So we combine MS, mass spectrometry. So we select a specific size. We use eye mobility to select a specific structure, and then we record this infrared spectrum. And with this information, we get the structural information of our species. And then we have to repeat it for every step in the aggregation curve. So in the end, we get actually the situation we have now. We can select very isolate specific Waldos under COVID conditions. So indeed, like I already said, combination is key. So we really need the combination of all the techniques together into one experiment. And at this moment, what we're doing now is we're testing two combinations. So we test, for example, what I would show first, we test the infrared with mass spectrometry and see what information do we get from there. So then maybe you have recorded already an infrared spectrum before, but most of the thing what you, I think, have done is taking an infrared spectrum of your sample in solution in your cuvette. And what you do here is you measure, if you would look at aggregation, at all the different processes together. So you might remember that, of course, when we do absorption, we have the Lambert-Beer law. When we measure the incoming beam, we have the outcoming beam. And if we take the ratio, we get a measure of how much is absorbed in our cuvette. Things change a little bit when we go to the gas phase. Well, first we write the equation slightly different. We talk about population and cross-sections rather than extinction coefficients, but they more or less mean the same. And we have still the same Lambert-Beer formula. We compare the intensity after you have the interaction and the intensity before the interaction of your light source. And then we use the absorption coefficient. And the length, of course, of your interaction is important. And this absorption coefficient is a measure of this combined with the population difference. And so the amount of population you have, the amount of ions we have in our mass spectrometer, times the cross-section, which is proportional to how much light is absorbed. So it's the probability that you have this absorption process. So if you're in the mass spectrometer, the question, of course, is what I would like to ask is, can you use the same approach, the measure absorption using Lambert-Beer, so by looking at the ratio between in and outcoming beam, to measure infrared absorption inside the mass spectrometer? Now, to answer these questions, we need some facts. So typically, if we measure an absorption, we want to have sigma of alpha L, which is the same as sigma Ln. We want it to be, the absorption, we want it between 0.1 and 1. So typically, say we have an absorption cross-section, what is a normal for, well, this is tryptophan for this molecule, it's 10 to the minus 20 cubic centimeters. Well, the length of interaction, say it's 1 centimeter. Then you need to have 10 to the 19 to 10 to the 20. So if you just take this red equation, you can see we need about this amount of ions in our mass spectrometer to be able to measure a decent absorption. But then a mass spectrometer, what you do there is you have your molecules that are in solution, you bring them into the gas phase. So your mass spectrometer, you have a gas phase environment. So let's say when we have 1 cubic centimeter, so when we have 1 centimeter length of air, it has 10 to the 19 molecules. So that would be okay. But we are not in air because we are in vacuum. So mass spectrometer operates under vacuum conditions. So this is the mass spectrometer. You bring the molecules into the gas phase. And when they arrive in the ion trap, it is over here. And say you also have an interaction region of 1 centimeter. So if you're in the gas phase, you have, for example, has a pressure of 10 minus 8 millibar, and this is a very low pressure, with the operational pressure, you have only 10 to the 8 ions in here. And then it's even worse because ions are charged. So they can interact with each other, so they repel. So if you have too many ions, if you would have 10 to the 20th times of ions in there, they would completely repel each other. So that's not possible. So in 1 cubic centimeter, you can have about 200 times 200 times 200, so about 10 to the 6th, 10 to the 7th amount of ions. So these are completely different numbers than the amount of ions that we need to actually measure direct absorption. So what does this mean? And then we move on. So if we have still the absorption cross-section of 10 to the minus 20, we say we can only have 10 to the 7th ions present. Well, what does it mean for our length? So, because that's the only other parameter we still have. So you see we need 10 to the 12, 10 to the 12, 10 to the 13, so more than 1 million kilometers length cell. So of course this is impossible. So to measure an infrared spectrum inside a mass spectrometer, you have to do a different technique. So we measure the absorption not as a difference between in and outgoing beam. So we don't measure the influence on the light, but we look at how the light influences the molecule. So we do it reverse. So we can look at the difference in ionization. We can use different in quantum states. So look at fluorescence, so the infrared light is absorbed or the UV light is absorbed. Does the molecule start to fluoresce? Or, as very ideal in mass spectrometry, we can look at fragmentation. So we have 2 types of setups of this, and I will not go in all the detail of the setup. That would be very welcome and happy to discuss. But what we do is we go to this mass spectrometer to look at fragmentation, because fragmentation is just a change of mass. So the molecule, the ion, absorbs infrared light, and if it absorbs enough infrared light, the weakest bond will break and you get fragments. And of course, we are inside the mass spectrometer, so we can really nicely weigh the fragments. And that's exactly what we're going to do. So we have our molecule, we excite it with light, it starts to vibrate and vibrate like crazy. We add more light, and at one point, the weakest bond will break. And we scan now the infrared wavelength, and when it absorbs, you will have this process. So you will see fragmentation as a result of infrared excitation. So we have a parent, we form fragments. So in a mass spec, it would like this, and we have no infrared or it's off resonance, you just see your parent signal. But when the infrared light is on resonance, so the molecule absorbs, as you see in the clip, it starts to fragment. We form these fragments as you see in your mass spec, and we record the yield of fragmentation as a function of the wavelength, and we have a beautiful infrared spectrum. So it's slightly different than you're used to, but this is how we do things. And then we can measure an infrared spectrum of every mass present. Then final thing on the technique. So what is the infrared that we use? Well, you have a full infrared spectrum, and it really depends on the information you look at. For example, You can look at the OH stretch vibration. You can look at the CO stretch vibration. You could look at out of plane vibration, so you go down to far infrared, or you can look at really terahertz vibrations, where you look how the whole peptide moves collectively. So what do infrared region do we use? So for that, we go to our experiments, and we do aggregation on 3 types of experiments. We form the aggregates, so the clusters on the fly, so inside the mass spectrometer. We form them in solution, and then we bring them into the gas phase, and we form them in droplets. But like every technique has in common, we do mass selection and infrared spectroscopy. So how did we learn previously? So we looked at this monopeptide to see what is important. First, well, we can see in the mass spectrum, we see in pink the monomer, in red the dimer, and in orange the trimer. And we could see when we play with our conditions, we could make higher order clusters. So it's very nice, we can form the aggregates. What we also learn is about the structure, is that we find monomer structures, and we also found that the aggregate actually remembers the monomer structure. So that really helps the analysis. So you can first look at the monomer, and then you can start to predict what your higher order clusters will look like. And finally, here's an example of an infrared spectrum, and only focusing on the CO position of the amide bonds. And on top is the ugly FTIR spectrums of an aggregate. So that tells you where the aggregate CO is. So what we see, we have free CO, there are always 2 in our aggregates, and that's always come on the same position, highlighted in pink. And we have this blue CO, which is informed inside the hydrogen bond of the aggregate, and we see it nicely shifts toward where the full size fibrous group is at. So this is very clear that you see this indication, this shift is representing this parallel hydrogen bonds, parallel aggregate formation. So we know the technique works. Well, then we looked, so what is the best infrared region to use? Well, if you want to look at aggregation, the CO, as you see here, works very well. But if you want to learn about the monomer structure, CO is not useful at all, because in black you see the experimental spectrum, and in the colored ones you see the calculated structures, which are slightly different. So you have the purple one, we have the blue one, and the green one. So they are folded differently, and you see, well, from this we cannot distinguish by, for example, the linear structure or with the 2 structures in blue, because they look very similar in the infrared signature. But then, for example, if you move to the NH red region, there you see that the colors, actually that the purple one matches much better than the blue one. So we now know that we have this purple structure. We can also use the far infrared, that gives us more structural details. So we typically use this NH red region if you want to know the overall structure of our monomer. We use the CO region if you want to look at this beta sheet formation, and the far infrared, it's much more difficult to measure. We actually only use when we need structural details. So now I talked a lot about the methods. I would really like to show the first results that we obtained since I started here in October in the VU on Parkinson's disease. So here you see alpha-synuclein. We focused on the segment of this alpha-synuclein, highlighted in blue, what is actually known to actually trigger the aggregation. So we did this infrared spectroscopy in the mass spectrometer. So remember that we measure the fragmentation yield, and then we measured fresh, and we measured after 2 days. And then we were very disappointed because within this CO region, so there's 2 peaks, we couldn't see any difference of the formation of aggregate. So when we formed the sample fresh, and after 2 days, when we thought we would have aggregates form, we see no different signature at all in our infrared spectrum. So that was a bit disappointing. So either the signatures are not informative, but well, we just found out it did for aggregation, or we need an extra separation technique. And of course, what can happen if you would have the monomer singly charged, or for example, the tetramer with 4 charges, the M over Z, what you measure in mass spectrometry, so the mass over the charge, is for 1 plus, and 4 units 4 plus is exactly the same. So maybe we need a different technique to be able to see this. So let's go back to our overview. So let's now combine mass spectrometry with ion mobility. So therefore we did the first experiment in Santa Barbara, and now we do them here. This is a schematic of the setup, but what is more important, here you have the sample in solution, then you separate them by mobility. So like I said, but they separate by size, then we can select by mass, and then we detect what we have. So if we would have a monomer 1 plus and a dimer 2 plus, they have of course the same mass over charge, and ion mobility separates not only by size but also by charge. So if they go to this field, the 2 plus, the dimer is actually of course twice as heavy, but they also have twice as much charges. And because then because the charge is more sensitive, and you have the two dimers together that actually are not like 2 independent monomers, but they are a dimer with interaction, they arrive first. So we can select by arrival time the dimer over the monomer. So let's go back to the Parkinson peptide. So what we see when we have fresh solution, we have one major peak and something small at the bottom, and the major peak, as you can see, is cut. If we then look after 3 hours, we indeed see something happening. We see formation of this peak is increasing. And after 1 day, we really see that this one single mass peak that we measured the infrared spectrum of is actually consisting of much more than this one peak. So what we know from analysis, that we start with the monomer, but we quickly form trimers and hexamers. So this is really interesting that you cannot see with one technique by itself. And now we do this at the VU as well, so it's a different instrument. And we first actually started on ALS, another neurodegenerative disease, but we can do the same trick. So here you have the mass spectrum with the segment, and the mass highlighted with 1301 is indeed the mass spec, the signature of our parent ion. If we then select that, we can measure what we call the mobilogram, or the arrival time distribution, as you saw on the previous slide. And then you see we have multiple peaks again. So this one mass spectrum actually consists of a lot of different aggregates. And then we can look at each peak individually and then look back at the mass spectrum. And then you see we use the isotope pattern of our molecule. And when there's only one monomer present, 1 plus, the one with 1 carbon 13, of course, is 1 heavier, arrives at 1302, and with 2 carbon at 1303. So we know this is the monomer. When we would have a dimer, we would have, of course, we would have 2 of these monomer units when we have with 2 plus charge. So when we have 1 carbon 13, so 1 mass unit higher, we divide it by 2 because we plot mass over charge. So we have a peak at a half unit higher. So as you see here in the green spectrum, so we know this is the monomer. And if we continue, we can see we have the trimer as well. So this is just a fresh sample and measured last week. So we can really use this technique to follow the aggregation process. So then I forgot one thing. So we have now the technique that we can follow this aggregation process, but we always started from, well, we never looked at the time points. And of course, this is your aggregation structure. You start from the peptide to the protein, it starts to unfold, and it starts to aggregate. So this is very much as depicted here. You have an egg, the egg opens, and a chicken or little dragon is formed. But with molecules is not that's not true. As I said, if we have 1 droplet of peptide in solution, we have there thousands of peptides that can start aggregating. And of course, they never start always on the same time point. So we want to have something that actually selects a specific time point, because otherwise we have them all starting at different time points, really complicating our process again. So we still, when we select the time point right now, we're still having a slight average. So what we want to do now is to bring one aggregation seed inside one droplet and let that grow. So we can really follow the aggregation of one seed, so one curve at a time. Well, therefore we need to develop something completely different. So we need to find a way to put aggregates in droplets and bring these droplets inside the mass spectrometer. And that's one technique we are working on right now. So this is what it looks like in the lab. So we have this chip where we have an oil phase and an aqueous phase with our peptide on it. And if you bring this oil phase and this aqueous phase together, you get this formation of droplets. And then in each droplet, we ideally have one aggregate formed. So what does it look in your mass spectrum? Well, rather than like a constant signal what we normally have, we have now spikes. So every peak means the arrival of one droplet, and then you have the oil coming in, one droplet, etc. But we can still then look average, for example, over 10 droplets and see the mass spec, or average over one droplet. So the nice thing is that you already see we have some aggregates present. The other thing is that we really have to work on the signal to noise, and this is something we actually are currently working on. So this brings us that we have droplets to select the time point, then we combine the eye mobility with mass spectrometry to select a specific conformation, and ideally we then have the infrared adding to our instrument and then measure. So this is the new lab at VU. Well, this is actually the picture of December. If you would come here right now, and of course you at one point welcome to have a look if you're interested or for an internship. Now we have a completely full lab with equipment. So for the last 5 minutes, I quickly want to highlight two of the other projects that we can use with this technique. So one is to control aggregation and one is on the diagnosis. So to control aggregation, so to stop from the, for example, from the formation of dimers to tetramers, or tetramers to octamers, we have these small molecules, levodopa, what is actually known for Parkinson's to actually have symptom relief, and ambroxol is a molecule depicted here that's thought that actually there's an effect on the plaques, but that's not really proven. So in collaboration with Samantha Hughes from the Han Biocenter in Nijmegen, we work on C. elegans, that are small nematode worms that are overexpressed with alpha-synuclein, so the protein for Parkinson's. So these are C. elegans that have Parkinson's disease. And what we look at is to look at their movement. And you see here when you have the wild type, it nicely moves over its plate, looking for food, for example. While the one that's Parkinson's, you can see that its movement is already affected and is much less flexible. Another one is that we will look at plaque formation. So we have a fluorescent tag to this protein, and we can really see, well, L4 is when they're young, and so the lifetime of a typical worm is 2 to 3 weeks. So you see that after 1 week, we really have this plaque formation highlighted by these dots in the head. And if you have a 2-week worm where it is close to its end, you see that these plaques are not even spots anymore. They just covered the whole outside of the worm. So we can test these small inhibitors by looking at body bands and plaque formation. So we checked levodopa and ambroxol. So here are the graph of average body band. So you have the healthy one, you have the mutant. So you see the reduced flexibility. When we then add levodopa, which is indeed known as symptom relief, we see that the movement actually is brought back to its healthy condition. So actually that's very interesting. When we would look at the plaque formation, we can see for the control, so it has no drug, you still have the plaques. And if you go to the levodopa one, you see all the spots. So it doesn't affect plaque formation, so that's still there. But for Ambroxol, you can see that the spots are all gone. So actually, Ambroxol indeed affects plaque formation. So that's very interesting. And these are just a study of, at this moment, 20 worms. And now we, of course, scale up this study. Then finally, how we use this technique for diagnosis. Well, this study started with Professor Perdita Barron here on the left, and Joy, the wife of a Parkinson's patient. So, and she claimed that she could smell the Parkinson's disease. She could smell that. She noticed that the smell of her husband changed over the time that he had Parkinson's, and even before he was diagnosed. So they started a small clinical trial. So they had a small group of willing people, with 6 people that were positive on Parkinson's and 6 that were negative on Parkinson's. They let them wear a t-shirt, a simple white t-shirt, let them wear it for 48 hours, and then she would smell blind all the t-shirts, so she didn't know it was positive or negative ones. And then we would test also with mass spectrometry. So the nice thing is he identified 5 of the 6 people that had Parkinson's. So she had 1 false negative, but she also had a false positive. So one that was not diagnosed yet with Parkinson's. And she said, well, this person has Parkinson's, and this person actually was later diagnosed with Parkinson's. So that was very interesting. So the smell, but of course the smell is just a change of your odor also contains of molecules. And if you have molecules, you can identify them with mass spectrometry. So what we did is we took swabs of sebum, so the fatty part of your skin, and we took it from the backside, actually a little bit lower as depicted in the picture, as it was the least infected by body cream, shampoo, etc. We collected samples of over 700 hospitals, so most of them in the UK, partly also in the Netherlands. And so it was a big team. And well, we measured, we had to analyze, so it's a team of mass spectrometrists, data analysis, neurologists, and we had all these samples, and we had them on this gauze paper to actually rub the sebum from the person. And then, of course, we had to take the sebum and the molecules from the gauze into a solution so we could use it. So there was a lot of preparation steps that we had to do to dissolve, well, put the gauze into solvent, extract it, filter it, extract it again, then dry it, redissolve it so we have the ideal concentration, and then it would be ready for any type of mass spectrometry experiment. And I will conclude with showing what we got. So we had this first study where we had about 140 medicated Parkinson's patients, 80 ones that were diagnosed but were not starting any drugs. And of course, we had the control group, and then of course we had to look at if they are a smoker or the alcohol intake or the gender, if they would actually influence the sebum, not the concentration, but the content. And we used there 2 types of mass spectrometry, gas phase. So we actually, when we look at the volatile, so you could actually, what Joy smelled, we could put in the mass spectrometer. We use liquid phase separation mass spectrometry for the lipid-like, of course, molecules. And of course, to compare the controls via patients, we had a lot of data analysis methods. So the nice thing that we found is that we found about 85% that we could really classify based on the biomarker profile if the person had Parkinson's, yes or no. And we really find on the LC work that we had we could find lipids that actually are could be potential biomarkers. So that's, of course, we now continue to work on, and now using the combination of eye mobility, mass spectrometry, and infrared, as I presented to you today. So to sum up, I hope you see that the combination of many techniques really bring new technology to understand aggregation, its mechanism. We can use it to find therapeutics and find biomarkers for disease. And one thing is that the infrared combination really brings the molecular specificity, so the identification, to a technique that is very selective and sensitive as mass spectrometry. So this combination really helps us to bring this knowledge. And of course, we will now apply, when we have developed the knowledge, to all the projects I presented you today. And before I really end, I would like to thank the whole team. Of course, we have the group, the MS Laser Lab at the VU in Amsterdam, shown here in the middle, with the collaborations that are very important, and of course also the whole division of bioanalytical chemistry. And yeah, I'm looking forward to have some discussion on the things I've presented, and please feel free to ask any type of question. So, thank you.